Thank you very much to all of you and for the opportunity to be here. First of all, I would apologize myself for my bad English. I hope you can understand me and follow the slide in the meantime I'm talking. As you can see, I'm working in Milan in a private foundation for pharmacological research, and I would like to share with you my experience in using NGS technology from a pharmacological point of view. When I started more than 20 years ago working in my lab, in my scientific experience, the idea was to approach cancer from a generic point of view, what is now called cytotoxic drug. Now, over the last 20 years, there is a paradigm shift from a generic approach towards a stratified idea, moving forward to the idea that it's possible to match a tumor with the best target to treat itself. Well, basically, all this philosophy is based on the great knowledge that we have got over the last years on the Human Genome Project, and what we have found that within each tumor it's possible to identify some genetic lesions that sustain and support tumor growth and its progression towards malignancy. So the idea is to match each of these genetic lesions with the perfect target to improve patient outcome. However, working on this field, there are many different issues and challenges that are emerging, that are growing up, and which hamper the possibility to improve the clinical outcome for patients. And one of the points I would like to discuss with you today is related to the issue of tumor heterogeneity, which is very important in one of the diseases I'm talking to you about. It is high-grade serous ovarian cancer. High-grade serous ovarian cancer is a really complex and heterogeneous disease from a clinical point of view, anatomopathological, etiopathogenic, and molecular point of view. For example, the TCG Atlas identified only 5 different molecular subtypes lagging behind a single definition of high-grade serous ovarian cancer. So the main issue in ovarian cancer from a clinical point of view is briefly summarized in this cartoon. All patients are debulked by surgery at the beginning. They're treated with platinum and taxane chemotherapy, and then in 90% of the cases, they relapse within between 1 year, 6 months. We can predict from the biological information the time at which this patient can relapse. But the time is important because after 1 year, they can be retreated with platinum-based chemotherapy, and they are considered sensitive to therapy. If they relapse within 6 months, they are resistant. And the question is that all the data that have been reported in literature are focused on the molecular biology of the ovarian cancer, or the tumor growing in the ovary, while we miss most of the information on the biopsy of the tumor at relapse after 1, 2, or 3 lines of chemotherapy. And from our studies, for example, since high-grade serous is a systemic disease with many different synchronous lesions at time of diagnosis, we observed that dissecting the molecular feature of ovarian cancer from NGS approach, so looking at a single nucleotide variant, we observed just that less than 5% of selected genes share the same single nucleotide mutation in the synchronous lesion. And in matched patients, when we analyzed their metachronous lesion after chemotherapy, we observed less than 2%. It means that this is extremely heterogeneous, and the biology of the tumor completely is a dynamic event that completely changes over time. So this is the reason why looking at a single biopsy at time of diagnosis is not enough to look how the tumor evolved and if you would like to introduce a personalized approach for this kind of the disease. So in this scenario, the use of circulating tumor DNA is found to be an optimal tool to improve our knowledge on how the disease is moving, how the disease is growing. So the second issue I would like to highlight to you is that in ovarian cancer, target therapy is, from my point of view, a missing step in the evolution. We have still the gold standard is platinum plus Taxol for first-line chemotherapy. Then 10 years ago, bevacizumab anti-angiogenic agent was introduced into the clinic, and now we have the PARP inhibitor which are looking for the BRCA1 and BRCA2 somatic and germline status to predict if some patients are eligible for PARP inhibitor treatment. So it means in this complex and heterogeneous scenario in which at least we have two single biomarkers, that is BRCA1 and BRCA2, as possible biomarker to predict, to know if this patient can be treated or not with some drugs. The advent of NGS technology is sound extremely important for introducing BRCA1 and BRCA2 analysis in the clinical management of high-grade serous patients, or also for monitoring healthy people for hereditary syndrome of breast and ovarian cancer. However, at least I can share with you the experience of the Italian situation and for also in the Europe. There is a great effort in introducing NGS into the clinical experience, while it's very well seated in the academic researches institute, but in the clinic there are many efforts to introduce this technology with no shared rules, not regulation on how in the clinical setting we can use this technology. And there is a great push to move forward into the clinical setting and the clinical diagnosis for this technology, while there are some great issues that are not still unresolved. And when more than 2 years ago, we decided to set up a diagnostic laboratory in our institute, we were facing with at least 3 main issues. The first was a fragmented workflow. So we needed to, if you would like to introduce NGS laboratory for diagnostic purposes for BRCA1 and BRCA2 in ovarian cancer, we were facing the possibility to have one single, very simple workflow without many different kits and solutions for many different components, and a complex to analyze and the data in the time frame of a diagnostic report and to control the cost of the system. So when we decided to set up this laboratory, the first step was to get ISO certification. That is, for a scientist, a really boring situation, believe me, with many rules to be written, to be followed, and every day. But these were the main issues that we were looking for. First, we were looking for a fast and robust sequencing approach, automatic or semi-automatic approach that could help us in receiving the ISO certification. The final point was, from my point of view, that all the data should be analyzed in a very automatic way with overlapping results from that reported by our bioinformatics team. Oh, sorry. So we looked for the GeneReader QIAGEN platform. That is a snapshot of our laboratory with all the instruments. And I would share with you some results using the BRCA1 and BRCA2 kit for germline and liquid biopsy. So we have selected this single platform for both of these situations. This is a clear example of 2 patients. For example, we found a germline mutation with a translational study. And we use these 2 patients as a proof of principle to validate the first step in germline analysis. The results were overlapping with the BRCA1 and GeneReader platform with those obtained with other solution. We repeated this with inter-experiment variability. This is a snapshot of the final report in which you obtained the same result. I skip this very quickly because I would like to share with you our experience using the GeneReader platform for circulating tumor DNA. This is a patient from which we have got the plasma at time of diagnosis, as well as the blood. At the first surgery, different areas were withdrawn for the patient in the operating theater. Then she received 3 lines of chemotherapy, and then she went for a second look surgery. And we got 2 different tumor biopsies. The patient was mutated in the germline for the BRCA2 gene, and you can see in every tool we observe the same mutation. Then interesting, when we look at the somatic analysis, these data reflect more or less what I told you before about the tumor heterogeneity in ovarian cancer. You can see different biopsies have a completely different somatic mutation in the BRCA2 or in BRCA1 gene. For example, in the peritoneum biopsy, there are no BRCA somatic mutation, while there were different mutations in the other biopsies. Then we look at the circulating free DNA, and we observed one mutation in the BRCA2 gene that was not present before chemotherapy, but we recruited them in a second look after surgery. It means that there was another lesion that was left by the surgery in the abdomen cavity that regrew after chemotherapy. While we observed here, in this circulating tumor DNA, this BRCA1 mutation that was present in one of the lesions we have observed. So this is just an important proof of principle study for us, showing that with one single solution we could use for detecting germline, somatic, and circulating free DNA mutation. So in conclusion of my simple talk, I would like to say that introducing this platform in our short experience, we have found a very simple workflow for data generation, for bioinformatic analysis, and finally is really cost-effective because it is in one single solution. So we received the certification, and these are the colleagues who are working with me in the Mario Negri. I would like to thank all of you for the opportunity to share with you my experience and my data. Thank you very much for your attention.
Sergio was nice and succinct there. So we have time for a couple questions. If anybody has interest in hearing more, has questions, gene reader questions. Not a platform that we're seeing as much as of in the US just yet, but it's coming, and we're going to see more of that. So, any questions? We got one here.
Ooh.
See, like, all these different kinds of mutations in the BRCA1 with different sites. What is the implication for the therapy that's selected? Does it change?
Well, yeah, there is, considering the mutation I showed to you are all, I reported all the pathogenic mutations. So all these are mutations that impact on the function of the BRCA1, of the BRCA2 genes. There are also some other non-pathogenic variants that can be identified, but all these mutations in the different area of the gene can impact on the therapy. So are all patients with all these different mutations can be selected for PARP inhibitor therapy. There is no, at the moment, for BRCA1 and BRCA2, is not quite different from TP53, which you have some specific mutation, or for EGFR receptor, which you have selected mutation for therapy. All pathogenic mutations that are considered pathogenic in BRCA1 and BRCA2 select patient for PARP inhibitor therapy. Thank you.